Okay, so the purpose of this lecture is to talk about how DNA makes copies of itself. If we go back to the cell cycle, you may remember that we had G1, S, G2, and then mitosis or meiosis or whatever was going to happen. And I explained a bunch of times that during S, a chromosome went from looking like this to making a second copy, and now it looks like this. And that this was a chromatid, and the chromatid was basically an exact copy of the other side. So DNA replication, what we're talking about here is what is the process by which it does that? How does it make the second side? That's the point of this lecture. All right. So the model that we believe is called the semi-conservative model, and you'll see why it's called that in a minute. I'm going to explain to you the basic experiment they did. You don't have to know their names, but if you were to be given that experiment, you need to understand what happened. So I'm going to show you the experiment that they did in a second, and then we'll move on from there. So the semi-conservative model basically says, and this you should have learned in regular bio or honors bio, theoretically. So what this is saying happens, this is what you would have learned in honors or regular bio, is that if we start with, let's just say this is our original DNA. What we're saying is that original DNA would open up, unwind, and split in half. And this would be my original DNA here all opened up. And then what would happen is you have little nucleotides floating around, and they would come in, and if this was an A, what would attach to it? A T. And if this was a G, it would be a C. And so basically a partner would match up, and in the end, what we would be ending up with would be two strands, which would now be our two sides of the chromosome, and each one of those strands has one from the original, and then one that's new, a new match. Yes?
Where do the partners that come, like if you already have a T here, like all the matching ones, where do they come from?
They're literally floating around. So there's extra nucleotides available floating around in the nucleus, and then what's going to happen is, as they're needed, they're going to come in and be attached. So you can assume that there's a supply of those available. All right, so are we good with that? All right, real quick explanation of the experiment that they used to show that this was the model, and then we'll go from there. All right, and I can come back to this after if you need me to. So here's what they did. They used E. coli bacteria, which still has DNA. It's a bacteria, the DNA is not in the nucleus, doesn't matter. They grew it in a particular isotope of nitrogen, nitrogen 15. Now in this case, they're not necessarily using radioactive isotopes. The purpose of using isotopes in this experiment is because they have different weights. So they have E. coli, and the nitrogens in the DNA, which remember DNA has nitrogen bases, all contain nitrogen 15, a particular isotope of nitrogen that's heavier I should say, than nitrogen 14. So now we have this DNA sample, and the DNA sample consists of, among other things, nitrogen 15. If I was to take this DNA sample, and I put it in a test tube, and I ran it through a centrifuge. You know what a centrifuge is? Like they use it to spin your blood to separate it, and like heavier stuff sinks further down. So if they spun this in a centrifuge, it would sink to some particular level. And we'll just say this is the level it would sink to, nitrogen 15. Now, what they weren't sure of was when the DNA copies itself, does it do this? Does it look like this, and this is a template, and then a new DNA sort of looks at this one, maybe wraps around it, and then makes a photocopy of it. Now, if this was how it happened, this would be called conservative. This would be a conservative model, meaning the original DNA stays intact, and then a new DNA comes in and forms. If that's what happened, since the only nitrogens, the bases, like I said, that would be floating around would be nitrogen 14, then if they spun this in the next generation, they would get one band at nitrogen 15, and then they'd get a second band some other place that was at nitrogen 14, because this DNA would weigh a little less than the original DNA, because it has a different nitrogen that weighs a little less. Does that make sense? Mm-hmm. Now, if it wasn't conservative, if instead it was semi-conservative, that would mean that my original DNA would have opened up. I'm using the opposite color, which I shouldn't be doing, but that's okay. and then a new DNA would come in made of nitrogen 14 and attach to it. What would we get if we now ran this through a centrifuge? Where would the band be? It would be exactly in the middle because it would consist of one old strand and one new strand. So that's the basic experiment that they did. They basically took it, they let it replicate one time, and then they looked to see, do we get two separate bands or do we get a band in the middle? And what they got was a band in the middle. And so this is how they showed that DNA replication was semi-conservative. You don't have to know the cesium chloride. If it's there, you don't even have to write it. I'm not going to ask you what the chemical they used to separate everything was. Don't worry about that.
You're not going to separate heavy and light.
No, don't even worry about that. Here's the experiment, really simple. They grow it in nitrogen 15. They add nitrogen 14 nucleotides and let it replicate. Spin it in a centrifuge and see where everything sinks to. They knew that 15 would sink to all the way down here and 14 would be up here. So if it was conservative replication, they would get these two lines. If it was semi-conservative, they'd get a line in the middle. And here's their actual results. Here's our F1, and notice they got a line in the middle. Now, if you continue this experiment, and you do not have to understand the F2 and F3, but if you can visualize, if they let this keep going, there's still only light nitrogen available. This is going to separate again. This time, this is going to end up with a light and a light. And this one's still going to be a heavy and a light. And if we let it keep going, you're always just going to get the light ones, the 14s, and you're always going to have one band for the original 15-14 combination. So they kept the experiment going and showed that that's what they got every time. If you don't understand this part down, don't worry about it. If you understand that they showed it was semi-conservative, that you ended up with an old and a new and it floated to the middle, then you're golden. Okay? Okay, so now this is the part you probably didn't learn in regular or honors bio. Remember we talked about how one side of the DNA runs 5 to 3 and the other side of the DNA runs 3 to 5. The problem is DNA can only be built 5 to 3. So that means that when the DNA opens, because it doesn't just all open at once, it unzips. So that means when it opens up, here's our DNA opening up. This side, our 5 to 3 side, is here, and our 3 to 5 is here. Now, if this side is 3 to 5, that means that the new strand that's going to be built is going to be built 5 to 3, right? Does that make sense? You have to have a 5-3 to match it up. But that's not a problem. DNA can be built in that direction. In other words, there's a specific enzyme that will literally add bases down here to the 3'. That's not a problem. It can build in that direction. The problem is there's no enzyme to build it the other way. So in other words, there's no way to copy this side like that. Because if a primer attaches here, it would only be able to copy that way, and there's nothing there. So it's sort of like trying to read a book backwards. So this side, no problem. This is called the leading strand because it's actually going to copy pretty quickly. As the DNA is opening, this is just being built right along with it. The other side, what could happen is we could wait for the whole DNA to unwind and then build the other side. Sort of like if I finish my book and now I give it to you, now you can read my book. But what they do, or what your cells do, is actually faster than that. What if I read the first 5 chapters of my book and then I gave them to you and you could go ahead and read up to 1 to 5. And then I gave you the next few chapters. In other words, what it actually does is it builds the other side. It has to build it in this direction, but it just builds a little chunk here. And now as it continues to unzip, it builds a little chunk here. And it continues to unzip, and you get a little chunk here. And so these little chunks...
So it's like part by part?
Yes. And then at the end, an enzyme just comes and seals it up. So the other side gets copied in little pieces. It's called the lagging strand, because it's going to take a little longer. And the little pieces have an adorable name. They're called Okazaki fragments. Those are the little pieces.
Oh, this is the way
Yes, it is.
that makes sense.
Good. I'm glad. And then in the end, there's an enzyme that comes in and it seals up the fragments. And so now your two sides, your two DNAs are complete. So the leading strand copies as the DNA is unzipping. The lagging strand copies, and I have some pictures to show you, and I also have a video to show you that walks you through this. All right, so this diagram basically shows what I explained to you. Here's our DNA, and it literally unzips like this. The 3 to 5 side, there's no problem because it can build 5-3. So this is going to be your leading strand, and literally as this opens up, this is going to end up in one continuous match. This side is going to be called your lagging strand. What's going to happen is as it's opened a certain amount, and I think it's fairly just random, this just comes in and attaches, and now this copies. Now watch what happens as this keeps opening up. Now it opens some more, and we get another piece. And then it opens some more, and we get another piece. In the end, you're going to have an enzyme that's going to come in and replace all these little green things, these RNA primers. It's going to replace them with actual DNA. And now this will hook together. Now, I'm not going to test quiz you on the telomere part, but this is the problem with telomeres. When this leaves, this is an RNA primer, you now have 3 little bases here that don't have a match. Remember what I told you, it can't build this way. So there's no way to replace what's missing here. When the RNA primer leaves at the very tip, you can't replace it. So what ends up happening is that's going to get lost, and the DNA is now going to be shorter than it was before. That's why we have the telomeres, because they're just junk DNA to protect the important information, because the DNA is going to get shorter every time it replicates because it can't replace the primer at the very tip, because it can't build in that direction. This isn't a problem. When this one leaves, it can still copy from here to here, from here to here. That's not a problem. Yes?
Is that why you can get to an age where you can stop having kids? Is your DNA so
That's part of it. I mean, there's the whole aging process, and yes, your cells get to a point where they stop dividing. They're basically told, or they have a programmed death. But the other issue with having kids is that if a woman that she's 60 takes hormones, she'll ovulate. It's just that your eggs at that point are 60 years old too, and there's a much higher chance of a mistake happening during meiosis. But it is possible. I'm sure you can read about 50-year-old women and whatnot who have kids. I mean, I don't. First of all, you know, you're going to be the only 70-year-old taking your child to kindergarten, which is going to be kind of strange. But all right, I didn't need to record that, really.